I'd like to start by acknowledging country. So I'd like to acknowledge the traditional owners and custodians of the land on which we meet today, the Wurundjeri people of the Kulin Nation, and pay my respects to their elders past and present. I'd like to introduce Haque from across the road at the PDI. So Ash is probably better off to give it a little bit of background on himself than me doing karaoke here. So take it, Ash.
Thanks, Dylan. Can you hear me at the back? Yeah, cool.
You don't turn on,
Yeah, my Kylie Minogue mic is working. Is this? Okay, good. I'm Ash. I'm from the Doherty Institute on the other side of Royal Parade. I moved to Melbourne. I'll give you a very potted history in 30 seconds. So I moved to Melbourne just before the pandemic in 2020. Moved my lab down from Brisbane at the QIMR Berghofer, where we studied immune responses to Plasmodium and Leishmania parasites. I was there for 14 years, and prior to that, I did a postdoctoral fellowship at the London School of Hygiene and Tropical Medicine, and prior to that, did a PhD at Imperial and my undergrad at Cambridge. So I'm a Brit, I've become an Aussie, I've got 2 passports, and that allows me to stand up here today and talk to you. I'm going to talk about developing immunity to parasitic organisms, and my first slide is not meant to be an insult. I apologise, and I know that we're in the middle of a rental housing cost of living crisis. I acknowledge that. I know that many of us are struggling to get on that first ladder of owning your own property. But let's just forget that for the moment and consider those people who still live with their parents as parasites. Okay? Go with me on this. So, how do you become a successful young adult parasite? Well, it's all about resources, right? You have to be able to extract resources from your host so you can live, and that benefits you as the parasite living in that organism or with your parents. And yet, there's a fine balance here. If you extract too many resources from your host parent, it can trigger, and if you pardon the pun, you can trigger an inflammatory response, which might result in ejection from said host parental home. So when we talk about parasites, you know, they often develop a very intimate relationship with their host, but they're often very, very skilled. And we might touch upon the skills that certain parasites use to enable them to avoid this process. But ultimately, what we're going to try and understand is when the host has developed methods for ejecting or killing or removing that parasitic organism. So for the purposes of today, I'm going to keep things really simple, and we're going to talk parasites that are either very small in the top left-hand corner or very big. There are some parasites that live in some animals. If you take the biggest animal you can think of, like a whale, there is an animal, a parasitic worm, that could be meters, up to 30 meters in length. So big means very big. But small means both very small in size, but also I want you to consider that parasitic organisms could be a single cellular organism up in the top left-hand corner. So a parasite could be one cell in size, or indeed could be many cells, could be a multicellular organism. So here's a list, a very simple list. So I'm keeping things pretty simple today. On the top left, you'll see some examples of single-celled parasites. You may well have heard of Toxoplasma or Leishmania. Number 4 on that list is the one that we are going to focus on today, which is Plasmodium. the causative agent of malaria. And there are 5 species that can infect humans and cause disease. And one of them, Plasmodium falciparum, really is an agent of morbidity and death, and has been for thousands and thousands of years. Well, we are going to touch on worms as well, and we're going to understand how our bodies really fail to eject worms. I'm going to give you a small fact that I learned from a researcher, Richard Grenchis, who's a professor of helminth immunity at the University of Manchester. And he said that about 1 in 7 of the human population is infected with at least one worm. So that's a pretty successful outcome for the worm. And many individuals, children, adults, rich or poor, are infected. And of course, living in a highly industrialized nation, that drops away. But nevertheless, 1 in 7 is a very, very high proportion. And if one thinks about Plasmodium infections, even with our best drugs at the moment, you still have 250 million people getting infected and getting ill. But many humans, after that first wave of getting ill, continue to harbor parasites for many, many years in their bloodstreams. So what we're getting is this flavor of, well, actually, our immune systems aren't that fabulous at getting rid of parasites, because they are with us and continue to be with us for many, many years. And I think one of the pieces of information that I find compelling is when I look at the list of FDA-approved vaccines. I think there are roughly about 60 of them. They are almost all for infections with bacteria or viruses. And thank God that we are good at making immune responses and vaccines to viruses. The last few years would probably pretty crappy if we didn't have that ability to generate vaccines against at least some viruses and protect our communities. You've got to have, in any lecture about parasites, some nice pictures. And here at Haque, and I can see in the audience you have a researcher who's interested in toxoplasma. And here's toxoplasma taken as a rather glorified picture from a recent science piece. And people are interested in how to cure and remove these single-cell parasites from our bodies. And as I mentioned earlier, there are worms. And there are many types of worms. And here you've got a roundworm, a hookworm, Haque americanus. And this horrible beast could find itself in your intestines, could burrow into the surface layer of your intestines and feed there. So I think one of the major points I want to make, and it is an important point to take about the difference between those single-celled parasites and the big multicellular worms. Well, the single-celled organisms can often replicate within the host. So say you get infected with Plasmodium parasites, there can be this cycle of replication that goes on, which means that from the few thousand parasites that are released from your liver after a week after being infected, you may get billions and billions of parasites in your body that sort of subvert and cause some of that illness. Now, I'm not suggesting here with worm infections that you only ever have one worm, but largely, whatever your infectious dose was, however many worms you managed to swallow or eggs that give rise to those parasites, that is the number of parasites that you have in your body. So I wanted to draw that distinction, that you got one type of parasitic class, the small ones, that often go through lots and lots of replication, and you've got another type of parasite, the worm parasites, that often don't go through a lot of replication within your body. They might replicate elsewhere, and that's a generalization. But of course, there are similarities between these. In both circumstances, you can get tissue damage. In malaria infections, the severe ones, lots of different parts of your bodies can get affected. It could be your brain, it could be your liver, it could be your kidneys, it could be, damage to your spleen. It could be anywhere. And the same thing with worms, and I'll probably focus on intestinal worms, but again, there could be some mild level of, or more severe damage if you get heavily infested with these worms. So some similarities and some differences. And I've touched on the fact that maybe our bodies aren't very good at removing or clearing parasites. But there are still things that your body can do. So for those small parasites, you could kill them. Maybe your immune system could kill them. If you could stop that replication process, that would also be very beneficial. But with worms, well, some of the things that we know as researchers our bodies can affect are either the expulsion of worms that are in your intestine, for example, or maybe kill the worms. And we're going to touch upon some of those mechanisms. And importantly, maybe also repair wounds, those macroscopic damage of epithelial surfaces that may occur as those worms bury into the lining of your gut. So the next slide is really the sort of slide that many lecturers might spend weeks on telling you about all the various mechanisms that our bodies may utilize to get rid of parasites. And I decided not to give that kind of classic lecture. As important as it is, I want you to think about themes and generalized aspects of parasite immunology. So there are some very common themes between the top left and the bottom right. We touch upon these molecules. Many of you in the audience are immunologically expert. You know what you're talking about. And so cytokines, these soluble signaling molecules that enable immune cells to interact and communicate with each other, are critical. And it may well be, and it actually is the case, that different types of molecules are needed to help your body mount one type of response to get rid of a small parasite versus eject a very large parasite. That seems sensible. Your immune system has to have that flexibility. So you might find that interferon gamma or TNF are important for these small parasites, and maybe IL-13, for example, could be critical for antiparasitic immunity. We can touch on those differences. You can see that there are also antibodies on both sides. And I've written IgG at the top left, and I've written IgE down the bottom right. And that's because antibodies are critical for protecting you against some parasites. For example, we know that IgG can coat the surface of free malaria parasites, and we think it can stop them transiting from one red blood cell to the next red blood cell. So that's a wonderful example of stopping replication. But we also know that IgE can coat the surface of certain large parasitic eggs or organisms, and that can facilitate other immune cells coming to the party to help kill those organisms. So, for example, I've written here a range of cells, immune cells, that are often called granulocytes. When you look down a microscope, they have granules in them. And once a parasite is coated in antibody, some of these cells can be attracted towards that IgE-coated surface and release granules and help kill that organism. And similarly, for smaller parasites, macrophages, which can engulf and eat these smaller parasites, might play an important role. And then really the focus of a lot of this talk today is going to be on a type of T cell. It happens to be the area that a lot of my lab focuses on, so we're going to sort of shift towards that. But you can see at the top left, I've written T helper 1, and the bottom right, I've written T helper 2. So we're really going to touch on that because back in the 1980s, and to signify that, I've put up something which some of you may have never seen, is a magnetic tape. This is how we used to listen to music, guys. We used to slot it into the car. We used to make playlists for our friends or special friends. And this was how we existed. And back at in that same glorious era, we also had a way of categorizing the type of immunity that T helper cells could mount. So I've written CD4 T helper cell. And we knew that they could turn into at least 2 different types, T helper 1, T helper 2. And for the sake of simplicity for this lecture, you might consider that Th1 can often be very helpful to kill or stop replication of small parasites like Plasmodium. And T helper 2 produces a lot of those cytokines and antibodies that could help expel or kill large multicellular parasites like worms. We are not going to touch upon the negative aspects of Th2 biology today, but it's important to understand that some of those same mechanisms are responsible for scenarios in which the immune system fails us, where we get diseases like allergies or certain autoimmune condition, but particularly allergic responses to hay fever, pollens, or dust mites, for example. Some of those asthmatic diseases and allergic diseases are also mediated by some of these processes. So that was the paradigm. That was what we worked with initially. And as we fast forward to a slightly more modern era, of course, we listen to music in a slightly different way these days. One can see that there's more complexity in the way that we regard these helper cells. And they are critical. They do help and help orchestrate all of those immune responses I've talked about. And in addition to those Th1 and Th2s that I've placed on the left-hand side, we have a variety of different other types of cell that can develop during an infection. And I'm not going to go into those details for those other types, the IT regs or Th17s, but I'm very happy to discuss those. The TFH stands for T follicular helper cell. We now know that these cells are really important for interacting with and educating B cells and facilitating the production of antibodies that can be so helpful in many instances. And in the next part of the talk, and I'm going to give you a small break so you can stretch and, get your brain in gear for the next section. We're going to talk about malaria, and we're going to talk about Th1 and TFH. So you've got 15 seconds break. You can do whatever you like. You can stretch. You can move your body around a bit. But we're going to conceptually change now from that small and big idea of parasite immunity through to thinking about malaria. So Plasmodium infections in which the parasite invades, starts eating within the red blood cell, and at some point replicates itself from one parasite to maybe up to 32 parasites before bursting out and then infecting the next wave of red blood cells. Now, we know that Th1 cells and TFH cells can both play a role in controlling that infection. Th1 can help activate macrophages to gobble up some of those free malaria parasites. And TFHs are critical for producing antibodies that might stop parasites from getting from one red blood cell to another. So both important, but if I was going to bet on anyone, I'd probably say that this process tends to win out. This is the one that's most critical. And that'll be important for some of the talk. So here you have a process where a T cell, a CD4 T cell, starts off with no function. I like to use the educational analogy of great potential in a primary school student, but no real skill set yet in vocationally speaking. And so how do they acquire skills? They get activated. And this happens within the spleen for a malaria infection. You can see blue B cells, concentrated areas of red for T cells, and there are some malaria-specific T cells that we were studying in green. But you can see that this is the theatre of education, much as you are here in a theatre here, hoping to receive at least one nugget of new information to take back with you to your research labs. So the spleen is much like a lecture theatre. Here you've got cells coming in, moving around, hoping to get antigenic education through what we call antigen-presenting cells. And conventional dendritic cells are primary in this process, and that's represented by this prism. Naive T cells slowly develop a range of different skill sets, Th1, Th2, Th17, Tregs, whatever. But that process is complicated. And this is why I really want to shift now from understanding parasite immunity, or those basics, to thinking about how we might research that immunity in this institute or our own or elsewhere around the globe. We know that as a T cell, getting from white to a variety of different colors requires a number of different biological processes that I'm not going to go into. That's not the point of today's lecture. But if a cell is moving from state A to state B, we want to be able to watch all of that process. And so as immunologists, we have just a classical way of doing it. But unfortunately, T cells do so, they get their education at different rates, just like some of us learn more quickly than others. We often all get there, but it might take us a different rate to get from having no education to that degree. So when we want to watch that process, we take snapshots, we take time courses, and we try and watch what those cells are doing. And that's the same as having a longitudinal clinical study where you assess the same patient multiple weeks, or you might have a mouse experiment where you take multiple time points. Now, at each one of those snapshots where you take a time point, you'll see that your cells are in a variety of different states, and they might be quite broadly distributed in the middle of that process because of some cells going faster than others. And of course, if you've got multiple states that are emerging, like I said, there's a Th1 and a TFH state emerging at the same time in the same organ of your body, well, it becomes really hard to deconvolute all of that. And so really what I want to talk about is the importance of assessing immune cells in as much detail as you can. And some of you would have done wonderful high-dimensional flow cytometry where you can look at tens of markers on your cells. And that's really helpful for understanding where a cell might be along this dynamic process. And that's what I've depicted down the bottom. Wouldn't it be great if you could assess cells in minute detail, every single facet of their character? Just as I would accept that all of you are different, and some of you might be really great at chess, and some of you might be really good at cooking muffins. I want to look at that detail of of immune cells, and that's really what we're going to talk about in the next section. How can we study cells in that level of detail? Quick stretch. Many of you will have done flow cytometry. It's been around since, the 1960s. I think I can recall a science paper showing some of those initial flow cytometry hardware. And it's wonderful because you can, in high throughput way, look at thousands and thousands of cells and look at expression of one molecule versus another. And that's sort of how we might have done things 20 years ago. But of course, as you know, things are much better at high dimensions. The more dimensions you can assess, the better. I don't know that I ever saw Jaws 3D in 3D. It was a pretty poor film, even in 2D, if I'm going to be honest. But you know, with the advent of certain technologies, I'll give an example like mass cytometry. It's a hybrid version of flow cytometry and mass spec. You could look at 40 different proteins on the surface or inside individual cells. So that's a lot of detail. And certainly that's what I want you to take home, is this idea that there's lots of technologies out there to look at in a high-dimensional manner at immune cells. But my own lab was interested in a technique, this is back in 2013, called single-cell RNA sequencing, and it's really become an industry tool for us. And what I like about it is it has a predictive aspect to it, if you like. So by looking at messenger RNA within a cell, you can make some guesses about whether that cell might make that protein, and also you can use that data. And here we're talking about 20,000 genes potentially from one cell, of what state that cell is in, whether that cell really was better at making muffins or playing chess. And many of you will have seen the same cartoon where now, with a somewhat of a monopoly, to be honest, from this company, you can isolate single cells. And that one cell pings up from the bottom of that slide and gets captured in this droplet with another reagent that comes along for the ride. And you can do that for thousands of cells, and you can start to isolate a single cell, look at the transcriptional repertoire, which we call the transcriptome of that cell, and start to understand in great detail what that cell state might be like. Now, that produces a lot of data. You might do a standard experiment these days, not a particularly huge experiment of 100,000 cells and 20,000 genes. And I was trying to be a bit cute. It was so big that it wouldn't fit on my slide. But it's a lot of data. And so we've been reductionist biologists for a long time, and we're sort of being pushed into an era where we're trying to be more systematic or systems biologists, and in fact data scientists, really, where with the right amount of computation we can take that huge Excel spreadsheet of 100,000 cells by 20,000 genes and get some biological inference out of that data. So this is just a simple example where we took one grey cell that happens to be specific for malaria, and we injected it into mice, and we looked at those cells at various different time points in different colours over the first month of an infection. And we can watch how those cells opt for either that antibody helping state or that macrophage helping state. And each circle, I should emphasize, is the transcriptome from a single cell, and there's a gray line, which is essentially a computational mapping process to try and stitch together the dynamics for every one of those 20,000 genes. So it's a useful process because you can then generate a resource where you can look at your favorite gene, and you can try and see when it's expressed in that model system. And you can look at any variety of genes. So it's really a hypothesis generator. And that was in some ways a dirty word in biology, a dirty phrase for a long time, where you wouldn't necessarily want to do an experiment that had no hypothesis, that it was about the opportunity to generate a hypothesis. But I think some of us are becoming slightly more okay with that type of experiment, as long as it feeds on into helping you generate new hypotheses. So we had a hypothesis here on the right-hand side that I'm not going to dwell on, but I want to highlight one important aspect about T helper cells and their ability to combat this parasite or that parasite, is that each T cell, when it's naive and sitting there in your spleen or your lymph nodes or rushing around your bloodstream, it has a unique receptor. Many of you will understand that in great detail. It has its own unique T cell receptor. And what I'm trying to indicate here is that that receptor, in a sense, gives each T cell a pastel-shaded propensity towards to go one type of skill rather than another. I like to think of that as, whether you want to become a barrister or a solicitor or a scientist or whatever. And so that's really important, because the data I showed you was a very artificial scenario with one type of T cell that was engineered, a TCR transgenic cell. But we're in an era now where, as well as looking at the single cell transcriptomes, which is the dots, there are ways where you can sequence the receptor as well. And that's fairly common. So you can now look at these black lines. These black lines are drawing together siblings, cells that came from the same original mother cell, if you like. And that allows us to explore how cells make, if you like, choices between becoming TH1 or TFH or ITreg or memory this or memory that. And that's the sort of technology that, as T-cell immunologists, we find quite useful. But I'm not going to show you any particular data on that. It's really just to help you understand that that's a sort of standard research technique in the field. So, quick stretch. And I think we're going into the sort of last phase of this talk, and then I'm going to open up for questions. And we're really talking about the importance of location. Location is really important in immunity to parasites, but really immunity to almost any organism. And what do I mean by that? Well, I showed this image of the spleen earlier, and you can see that it's complex. There are areas where they're rich in B cells. There are areas that are rich in T cells. There are areas in between them. And that is just a simplified version of one of your secondary lymphoid organs. Your lymph nodes are very similar in many respects. There are some unique differences that we're not going to talk about today. But the way that immune organ is constructed has a really, really important role to play in making sure that immune responses can be mounted, that they're controlled. And so it's really that aspect that I want to talk about in this last part of the lecture. Wouldn't it be really good if you could both assess immune cells in that great detail that we've talked about, but also look at where they are at the time that they were doing whatever they were doing? Now, that's really important, because if I wanted to track how you won that Nobel Prize, I'd love to be able to see how you're interacting with other really, really important people in your life. So it's in the same way for watching what shapes a human life, it's really quite important to look at all the other cellular partners that might influence what happens to any given immune cell. So what we're really talking about is looking at the spatial location of immune cells in this instance, and how that might govern immune responses to parasites. So the term that we use for some of our work is spatial transcriptomics. And this is just one example of how one might do that. There are a number of ways, and WEHI has a number of platforms that are becoming established, maybe more established than others, but there's a suite of them that are coming online where you can look at as many genes as you possibly can in a cell, and as well as having a coordinate for where that cell was in the tissue. So what I'm showing you here is a standard sort of microscopy slide with some sort of bead array on top of it. Now, those beads are positionally sequenced, so you know where that bead is. You know the barcode of that bead, if you like. And that bead, one of those beads, will capture messenger RNA when you slap some tissue on top of it and perform the protocol. So you get localized messenger RNA capture across the entire array. So that messenger RNA is sequenced, and then you have data which tells you what the messenger RNAs were for that gene, and then where it was in the tissue. So here's some data from us trying to use one of those arrays, and that was a couple of years ago, but it's just to give you a flavor. Let's say each one of these violins, of the 8 violins, represents the data from one slide, and we had 2 conditions. In gray, we had an uninfected scenario, and in pink, we had a scenario in which the mouse was experiencing a malaria infection and had done so for a week. And on the y-axis, what you see on the log scale is the number of genes, the number of mouse genes detected per 10 micron bead. That's one of those beads was 10 microns. And so you can see, there's a big range, but we're talking hundreds to maybe a few thousand. And that data, when you then start to use that data to color each bead according to the genes that were present or detected, you can see, well, something that looks rather messy, but it's telling you that those beads that have the same color had the same kind of repertoire of genes captured. And for those of you who are immunologically inclined, you might be persuaded that there's some evidence of immune structure there. And then when you start to look in, uninfecteds on one side and infecteds on another, and the colors don't matter, it's the general point that you can start to capture microanatomy without having to do anything else to that data. Just harvest genes, and you'll see structure, and you'll see changes in structure. You'll see areas that are rich in newly generated antibody secreting cells like plasmablasts in one area of the red pulp, for example. But of course, that's a very strange technology, because if you've got a fixed array and 2 cells land on top of that bead, it's going to capture messenger RNA from 2 cells. And so we're currently left with the problem across the whole array of how do we deconvolute that into single-cell data. And so the current method, and it's imperfect, and I would argue that it's a common problem across each one of these spatial transcriptomic methods, is that you have to merge your map, your ST data on the top with some sort of ground truth data set, a data set is single-cell data. And then you can start to essentially convert each bead into having a proportional contribution from one cell type or another. And the process happens to be called robust decomposition of cell types. And so you can start to look in great detail, and you can start to use techniques to look at co-localizations of cells. And this is something that one of our PhD students, Cameron, simply I highlighted an area of a certain size and then walked around his puck, as he calls it, and looked for evidence of this cell being close to this cell. And you can generate data that have heat maps and correlations where, in a semi-unbiased way, you can have as many different cell types as you have single-cell references for, and start to look for, in red, evidence of co-localizations. And we were interested in whether these TFH and TH1s, which are necessary for that malaria immunity, were located near certain cells. And one of the cell types that we were interested in was whether TH1 cells were actually close to monocytic derived cells. And so this data suggested that indeed it was the case. So red and yellow at the top were co-localized, whereas there was a control scenario where yellow TFHs were not co-localized with monocytes. But it starts to allow us to test for co-localizations and then go back into the wet lab and do some more classical experimental immunology where you might deplete a cell type or deplete a gene within a cell type. It also allows us to generate databases where we can look for inferred cellular communication or molecular interactions between two cell types. Based on the depth of that data, we can say, oh, I think maybe these monocytes are interacting with those T cells via one of these inferred receptor ligand pathways. So I want to come back to parasite immunity, because now that we've really explored some of the research techniques that we find useful for looking at T cell biology during infection. And well, what's the point of this? Well, one is that we think that boosting TFH responses in this particular scenario can be helpful. Our mouse models show us that if you can accelerate and boost the follicular T helper cell response and in some cases drop the Th1 response, you can get mice to cure their infection that would otherwise become chronic in these scenarios. And one example that I'll finish with, and this last couple of slides, is a type of signaling pathway, a cytokine signaling pathway called type 1 interferon that signals through a receptor called the IFNAR1 receptor. Now, what we found is if we knock out that receptor, in green, you can see that T cells rush in towards these red B cell areas more effectively if that signaling pathway is not around. And in these graphs, what I'm really showing you is that in the absence of that signaling pathway, you get antibody more rapidly made, and that associates with a more rapid control of infection. And that happens in 3 of the different mouse models of malaria that we run. And so that's to say that our immune systems aren't fabulous at controlling these Plasmodium infections, but there is hope. And indeed, blocking that type 1 interferon signaling pathway was validated in some human studies, and it allowed us to trial it in humans. And so, in my old institute, we used a drug called ruxolitinib, which works in that pathway, and we've given it to, believe it or not, human volunteers who have been infected with Plasmodium. So you can do this for many, many different infections now. We have ethical approval and safety profiles to show that, well, in this case, 400 at least humans have gone through this process. And we're looking to see whether blocking that pathway can actually have a beneficial outcome in antibody production and control of Plasmodium infections. So there is hope for those, for in many instances, for us to be able to modulate immune responses to help us eject or kill parasites. But just to summarize, what I wanted to talk to you today about was the concept of parasitism. And indeed, the idea that there's really a fine balance here, and it's about resources. And that for many parasites, they are exceptional at walking that fine line. It's that case of, talking to a parent and trying to extract money, but not too much money, so that you can stay living at home and you can still have a good time living at home. So parasites do that very well. But what I'm hoping I convinced you of today was that there are mechanisms to kill and stop parasites. And I really focused on Plasmodium and malaria. But there are some stunning mechanisms that the body has to expel parasites. There's a wonderful one where IL-13 causes your gut epithelial cells to ramp up the rate at which they're recycled. So someone says it's a bit like an escalator. IL-13 switches up the escalator speed, and it allows epithelial cells to be shed more quickly and expel parasites. So there are mechanisms out there for expelling and repairing wounds. So our immune systems have gone some way to expel these parasites, but there's still many, many opportunities. And it's also worth highlighting that there's a rich history between parasitologists and immunologists. What I found when I came into this field was that there's a lot you can learn about controlling your immune system from parasites. So, for example, parasites might secrete a factor that helps calm immune systems down, and they do that frequently. And so if you can harness that, well, maybe you could treat some of these diseases where our immune systems have gone a bit haywire. Allergic diseases might be one example. So there's something to be learned about our immune systems simply by studying the way that they do or don't respond to parasites. So I'd like to leave you with that theme, and also to show you that we've touched on Th1s and TFHs, touched on Th2s. There's lots of other things that we can talk about if you so wish. And to end really with that, with a note of hope, this is a wonderful cartoon from 1802 in which James Gillray, with a bit of tongue-in-cheek, was suggesting that one of the negative consequences of getting the cowpox vaccine to protect you against smallpox was you'd start to produce small cows out of your skin. But that was obviously a hugely successful vaccine drive, and perhaps in the audience we have amongst you people who are going to help us generate the first real wave of truly efficacious parasitic vaccines. So with that note of optimism, I'll stop and invite any questions. So thank you.
Thanks for the really wonderful talk and all the beautiful images. So my question is, I don't have a very strong immunology or parasitology background, but I was just wondering, like for the observed improvement when we knock out like interferon receptor, how do we know that the observed improved response is due to the improved like TFH, and also due to the decreased Th1 rather than due to interferon signaling block?
So what I would say is that to conduct those clinical trials, association is your best friend. So they would look at titers of antibody, for example, in the serum of those individuals. So you'd look at how much antibody you've got, and you might also compare that with some other metric that you could measure early on. In this case, it might be something called STAT3. So STAT3 is a transcription factor that gets phosphorylated. You could look at the degree of phosphorylation of that and try and correlate it to the amount of antibody to give you some clue whether there's a strong relationship. And the variability amongst humans then becomes a very powerful tool to look at that, to imply causation. And then, of course, after that, you can re-challenge humans. Believe it or not, you can. There are also people who've generated protocols allow you to re-challenge and then look at the outcomes of those antibody titers. So I would say that I'll stick with clinical studies because that's the most relevant, really, and that's how you would do it.
And also, I got another question: is for the observed improved immunoresponses, is it specific for Plasmodium falciparum? Because I do know there are quite a few strains. Another one like the Vivax. So because I think they got some difference in their like parasitology or like. You're
right. So falciparum has a unique AT. Is AT rich to a greater degree than Vivax. So the Plasmodium genome is 80% AT. And which is super high, right? And so some people have suggested that there are certain motifs in the Plasmodium genome that actually triggers this response more so than Vivax. Vivax is very different in many instances. It can hide away in your liver. It doesn't cause the same amount of death as the other one. In some ways, you might even argue that it's a better parasite because it really treads that fine line to a slightly more without causing too much disease in its host. I would say that's a controversial thing to say, but there are certainly major differences.
Thank you for the presentation. I don't know the current prevalence of malaria in Australia, but I'm quite aware of areas in the world where we have high burden of malaria. So my question is, are you collaborating with such places as to your work on malaria? Thank you.
Yes, so we collaborate with a few different researchers. So certainly we collaborate with. So my current collaboration would be with members of the funded by the NIH who work in certain sub-Saharan African countries, particularly Uganda, and there are certain areas that unfortunately still get high seasonal exposure where children get bitten, tens, hundreds of times within the season. And so one of the things we're looking at is the impact of repeated infection on kids, because often we talk about immune responses being generated and then dying down and clearance of the parasite. But what happens if the child gets infected again before it's even had a chance to resolve? And so these are scenarios that we don't try and do any of those clinical studies on our own. We just simply wouldn't have the funding. So what we do is we stick with our modeling approaches and we collaborate to look at, in particular, longitudinal studies of kids in those regions. So that's just one example.
All right, thank you, Ash, for that amazing talk. I'm really curious about the spatial transcriptomic methods. Given that it's a relatively new technology, what do you think is the best platform for spatial transcriptomics right now? And what is the bottleneck of this new technology?
Yeah, well, I'd say, first of all, no company is paying me for any of these comments.
Yes.
I like the idea. It depends what you were trying to achieve. If you're simply looking for discovery, you want as much information at as high a resolution as possible. And so I tend to favor some of those ones where you don't have to design probes. I know that you can get whole genome scale probes for certain companies, but I like the ones where you're just sequencing-based technologies, and I like the higher resolution versions are also attractive. So I do quite like BGI's STomics platform. I know that you're certainly trialing it here at the Institute. The biggest challenge is actually how do you take your data and work out which cell. So you can go as high as resolution as you like, but at some point you're going to need to say that messenger RNA came from that cell. So what we call cell segmentation is an important area. And so it seems to me that having ground truth single cell data sets will remain helpful even when you have these subcellular resolution platforms. But I'll say that that's one that is attractive. Although we haven't generated any data in our group, we've used a Curio seeker, which is the bead fixed array, and that is also useful to us.
Thank you.
Awesome. Thank you so much for that talk. I'm also pretty interested in the spatial side of things as well. I was wondering if you know what is the ground truth, like how do you do the ground truth for like a sample you're running? How do you know which cell all those genes come from?
So when we started in those experiments, we would really take one piece of tissue and then divide it up for different protocols. So some of it would then get sliced for spatial transcriptomics, but we would in parallel generate a droplet-based single-cell RNA sequencing data set and then use that. But there are increasingly very large data sets publicly available. So say you had a piece of tissue from a certain human biopsy, you could certainly then go back to those huge data sets of millions of cells, multiple different types of tissues, and then use that as your ground truth to then interrogate your map. And that's really how we're going to go in the next. Yeah. And that's going to be the power of initiatives like the Human Cell Atlas is you want an atlas. You don't want to have to keep generating your own ground truths. They will be there for you. Of course, things change in any disease, so the applicability of those, it will be in question when you start to look at disease data sets where there could be some really weird cell types for which you don't even have the ground truth. So there'll be a caveat there, but nonetheless very powerful to have that reference.
Yeah. Awesome. I also had just another question. I was like wondering if there's anything you found through spatial like transcriptomics that you think you might have missed through like kind of traditional experiments.
Yeah, so we thought we can see two cell types that will remain nameless that are very close to each other. That I can't see any papers discussing possible co-localization or interaction. So we're in the middle of trying to validate that with classical microscopy, and then the other classical approach is then to deplete one cell type and see if it impacts upon the other cell type. So yes, we feel it has been useful. The other real use is when you've got cell types that are really hard to make single cell suspensions out of. So for example, the stromal cell network that has a really, really important role, well, they're really hard to work with. And so you can see those and map those using those techniques.
Yeah. Awesome. Thank you.
Thanks, Ash. That was really interesting. I was just wondering with the spatial transcriptomics as well, whether you can map the parasite genes on top of that to get a better idea of host pathogen interactions there, and whether you can correlate active infection or latency and those kind of things.
Yeah, you can. So we had a look. We mapped the Plasmodium shibaldi genome to those pucks, and we can see very clearly strong signals in the red pulp area as one might expect. The level of detail is quite low, actually, though. So to give you a flavour, for any given 10-micron bead, we could detect 4 parasite genes. So we can't really do too much with it. We haven't tried very hard to do too much with it, but there's certainly a possibility that you could pick up different parasite states. And I think it'd be awesome to look at maybe a leishmania infection where you've got a lot of really amazing foci, and you could really delve into some of that detail. So I think that would be cool. And actually, other technologies like the Stomix one also appears to allow you to look at not just polyadenylated messenger RNA, but other species as well. No, messenger RNA, prokaryotic. And so microbiome assessment starts to become possible with these techniques as well.
Yeah, they beat me to it with that one. But I was going to ask if you'd ever seen any of the pathogen transcripts with the single-cell RNA-seq approaches, so dual scRNA-seq, or is it just too lowly abundant to pick up?
We tried, just for Plasmodium, to look in phagocytic cells. Didn't pick anything up. Yeah.
Online. Nope. Okay. Any final questions for Ash? Good. Oh, all right. Well, thanks very much for the talk. Cheers.
Thanks, everyone.